So when you go back, you went further before the and then we
That's going to bring the volume.
Welcome back, everybody. I trust everybody had a good lunch. We're getting ready for an action-packed afternoon session. I want to do a few housekeeping announcements before we begin. This afternoon session does not have a long break period for coffee, so keep that in mind. Be comfortable, relax, but the break prior to the keynote speech at 3:30 is only 5 minutes long, so be back in time for that to start. Of course, please turn your cell phones back to silent. And I want to also announce that our first talk is being live video streamed. And as a result, we'll try and repeat questions from the audience because the audience won't be mic'd for this particular talk. And if not watching live, of course, this will also be presented on YouTube, and you can find the link to the YouTube broadcast of this particular talk through the ICCI website and also on Twitter at Vet School's handle on Vet College. So you can find this presentation at both of those locations. So I'd like to start with reminding people that in this symposium we do have local regional guest speakers. We heard from Dr. Wells this morning from Walsall Park. We had speakers from Waterloo, and now from McMaster University, I'd like to introduce Dr. Yonghong, who has been instrumental in the field of oncolytic biotherapies, which he'll talk to us about today. He's a professor in the Department of Pathology and Molecular Biology and is also a member of the Ontario Regional Biotherapeutics Program and the National Center of Excellence with regard to the BioCan RX, which is again the implementation of oncolytic biotherapeutics. He studies immunology and dendritic cell immunobiology and collaborates with investigators such as Dr. John Bell in Ottawa and others at McMaster like Dr. Oncolytic in bringing this into phase 1 clinical trials. Of course, here at the Ontario Veterinary College, we're also interested in applying this and have been successful in obtaining funding to take some of these concepts and clinical modalities into early stage trial in our technology as well. So please join me in welcoming Dr. Wen today.
introduction, and also thanks for the opportunity to share the work we did in the past few years, from our bench work initially to clinical trials. So it's pretty scary to be on the video. I've never done it before. So this presentation was prepared by my colleague, and Vicky. He was initially supposed to be here to present this. Actually, I like my version to tell the story that we could develop this process. But we prepare this, so to make the best to convey the information we put together. So you might have heard a lot of immunotherapy stories recently because some breakthrough using antibodies to block the check inhibitors, the lactic regulatory pathways, and some T-cell downward transfer therapies, which are very impressive. So I'll introduce a new concept to using replicating viruses to treat cancers, and we use that to bridge both oncolytic virus and immunotherapy. So the concept of oncolytic viruses is so almost all viruses could be selected or engineered to be selectively capable of replicating the tumor and disrupt the tumor to achieve the therapy. So the concept was actually inspired by occasional observations and stories in my here, chicken farmers whose tumor, through remission, when outbreak of Newcastle disease within chicken population, or kids who have lymphoma, and the tumor also not gone away after measles infection. So you may wonder why tumor cells are more susceptible to virus infection and being destroyed and left, the host, the normal cells undamaged. So it's because normally when normal tissue cells were infected by viruses, and those infected cells will stop translation, will either undergo mitosis, to sacrifice themselves in order to protect the host. So that's the way how we get protected. And also as a consequence, those infected cells will also signal the entire immune system to mount subsequent protective immunity against pathogen the virus infection. However, the tumor cells, because the deal they made with the devil, stop translation, they actually resist that because they just go on to proliferate, and also they try to evade from immune surveillance. That's how when tumor cells are infected by viruses, and they cannot stop the viral replication. So as a consequence, the tumor cells are more susceptible to virus-mediated disruption. So with the understanding how different between normal tissues and tumor cells, so we could actually manipulate the viruses more specifically infect the tumor cells. There are different ways to do that. So there are three typical methods we could use to achieve specific targeting of tumor cells. For instance, we could use the difference between tumor cells and normal cells, using some defects within the tumor cells to specifically allowing viruses to replicating within the tumor cells or not normal tissues. We could also use tissue-specific promoters to restrict viral replication within certain tissues, especially the tumor cells, to achieve specific targeting and disruption. We could also engineer viruses with some other components, maybe single-chain antibodies, to target certain receptors on cell surface to guide the entry of viruses into certain cells. So all those mechanisms will lead the normal cells infected. So for instance, we'll talk about the tumor cell defect. The pathway I will talk about later on is the type 1 interferon responses. So a lot of tumor cells, you can see that the examples listed here have lost the ability to respond to type 1 interferon. So type 1 interferon is the most effective way in antiviral responses. For almost all the clear cells can produce interferon of an viral infection. So that's our first line defense fighting against the viruses. Okay, so the tumor cells, a lot of the tumor cells have lost such an ability, so we may be able to use this difference to specifically target tumor cells. So normally here, for instance, so when a cell sends infected by the virus, and the virus will get into the cell, which will turn on the type 1 interferon, especially the interferon beta responses. So interferon beta will be secreted outside of the cells, and that will engage type 1 interferon receptor and stimulate more type 1 interferon production to amplify the process in order to control viral replication. However, the normal the tumor cells have lost such a capacity. That's why viruses can replicate within the tumor cells. So, such understanding led us, especially John Bell in Ottawa, first characterized the rabbit viruses, especially the VSV. The VSV virus, the first discovered as a prototypical rabbit viruses with anti-tumor or oncolytic capacity. So those rabbit viruses is a family that a lot of viruses that are extremely sensitive to type 1 interferon. So when they get into or encounter normal cells, they will be restricted by interferon responses. However, though, when those viruses get into the tumor, and they will have a chance to replicate to destroy the tumor. Now, in addition to such biological property in terms of a differential response of interferon between normal and tumor tissues, and there are some other advantages associated with the clinical application of those viruses as oncolytic agents. For instance, this virus is not human pathogen. It's very infected by the viruses. So we do not have neutralizing antibody, pre-existing antibody in the body, which will allow us to put the virus into the host. And then also those viruses were pretty easy to manipulate and can accommodate a large insert of genes and can be produced in hydrate, which will be the clinical lead.
So here's an example how we provide evidence that the VSV can directly infect the tumor cells. For instance, this is a normal mouse lung, look like it's smooth the surface. If we inoculate CD26, the mirroring colon-carcinoma cell lines, intravenously, so those the tumor cells will form tumor nodules on the surface of the lung. So after tumor formation, if we infuse virus through intravenous route, expressing GFP, the green protein, so you can see that the tumor cells were all lighted up by the green color, suggesting that tumor cells are not only infected by VSV, but the viruses are actually replicating within the tumor cells. As a consequence, so those tumors can be emulated by the viruses. Of course, we did a bit of more work to further engineer viruses to attenuate their toxicity, but still preserve their oncolytic capacity. So you can look at it, if we use UV to kill the virus, the dead virus didn't work. And however, the live mutant virus can result, the tumor regression. And if you look longer, those treated animals can survive pretty long time. Most importantly, if we re-challenge those cured animals with the same tumor later on, several months after, those mice could be still protected. Now, at that point, viruses were already gone. So apparently, the protective mechanism must be mediated by the immune system, suggesting this oncolytic viral therapy may also turn on immune responses to, protect the host for a long time. Now, the same actually phenomenon was observed in patients as well. So this is another oncolytic virus called TVAC, which is based on herpes virus. And this virus has been engineered to express a cytokine called GM-CSF, which can activate and recruit antigen-presenting cells such as dendritic cells. So the company, the Amgen, got these viruses and ran their phase 3 trial. And what they did was, if they just inject the virus into one tumor nodule, this is the melanoma tumor cells on the surface, on the skin, and you can see that after 6 weeks, not only injected tumor nodule was regressed, and the surrounding tumor nodules were also respond to this therapy. And more interestingly, the distal site, not nearly, close to this injection site, the tumor nodules were also eliminated after the months after treatment, suggesting again, in addition to de novo oncolysis, and the immune responses could be also subsequently induced by the therapy. So this is very encouraging, right? That the oncolytic viral therapy, you can achieve both the immediate destruction of the primary tumor, and they also may be able to generate long-term protection. However, though, when we start to test how broadly this effect can be applied to other tumors, we found it's very tumor dependent. For instance, when we use the murine melanoma B16 model, where we lock the tumor cells into the brain and treated with the VSV GFP as a mono oncolytic therapy. So you can see that virus, yes, indeed get into the tumor, express the GFP protein, and if you look at the tumor growth early enough, the 4 days after treatment, you could see actually tumor destruction compared to untreated tumor, suggesting that oncolytic component is indeed the work here. However, though, if you look at the survival, this early destruction did not necessarily translate to any benefit of prolonged survival. Okay, so now we learned the immune responses could, the immune system could contribute to this. As an immunologist, so we wondered whether we could manipulate, the viruses, for instance, here, further, where not to just rely on the byproduct of those viruses to occasionally, randomly, induce immune responses. Instead, we engineer viruses purposely with tumor antigens. So in that scenario, we could also, we could achieve oncolysis and what they call the in situ subsequent, endogenous immune responses and the virus-mediated antigen presentation in the periphery to secure the induction of immune responses. So that the idea that sort of the oncolytic vaccine was the intensive, discussion and the testing, by Baron Bradle. He is in the audience. He's the assistant professor here at the University of Guelph. When he worked in the McMaster and my colleague Brian Lichty. So we thought if we can engineer oncolytic virus to become an oncolytic vaccine, we may be able to achieve both the oncolytic benefit and also immune stimulation. So this how this sort of a two concepts combine together. So for the people who are focused on cancer immunotherapy, what they do is either use a vaccine to activate T cells in the periphery or put the T cells into the system where they hope those activated or transferred T cells will get into the tumor and to do the job. Okay? So oncolytic viruses, we just described their biological property. We can engineer them to particularly target the tumor. So we actually could, using oncolytic the virus, we conceptually could achieve, destruction of the tumor environment to deal with a lot of the problem which the pure immunotherapy cannot, right? And especially the oncolytic viral therapy can occasionally generate immune responses. So you may also benefit from host immune response. So we thought if we combine both the advantages where the T cells are, pretty functional and the viruses can disrupt the tumor environment to deal with the immunosuppressive or tolerogenic the environment, we may be able to achieve both the T cell activation expression in the periphery and the destruction of the tumor, and also the replication and inflammation within the tumor will recruit those activated T cells in the periphery into the tumor and sustain their activity in the tumor as well. So to test that idea, the oncolytic vaccine idea, so we engineered VSV, our prototypical oncolytic virus, to express a tumor-associated antigen called the DCT. DCT is a normal enzyme expressed by normal melanocytes involved in melanin metabolism. So it is a tumor-associated, but not really specific, the antigen. So it's a self protein. But DCT is overexpressed by melanoma tumor cells. That's why we could, take advantage of this differential level of the protein expression to specifically target the melanoma tumor cells. So this is our oncolytic vaccine, the VSV expressing the DCT. So what we thought, this oncolytic vaccine did not work as we expected. Right? There's no increase of any benefit of survival. So we wondered whether this vaccine did not work as immunostimulate agent. When we look at the T cell responses against the tumor antigen we put it into the DCT, that was very minimal, small response. However, though, if we look at the response against the virus itself, it's very strong. So the vaccine, the agent worked to stimulate the immune responses, but immune responses dominant against the virus, but not transgene. This is not entirely surprising because viruses are foreign, are more immunogenic, right? So the immune system are supposed to, do not respond against the foreign, not self, the response against the DCT. So that's where we wondered, so how can we get away from such immune, evolutionary setting, right? Because the immune system are supposed to do things like this. So if we really want to achieve anti-self, anti-tumor, the response, the weakening immunogenic response, we will have to reprogram the system, reverse this setting. So that's where we come up sort of about the prime boost, the regimen idea, where if we use a different virus expressing tumor antigen to trigger the response against the antigen first, and then followed by our oncolytic the vector expression the same tumor antigen. So now you have a memory. The immune system has, anti that specific transgene the response, and the subsequent response will focus on that transgene to avoid anti-tumor response. Nonetheless, the viruses will get into the tumor and will, have direct debulking effect. It will recruit the cells into the, the environment. That's all our fantasy, the imagination after we analyze the situation. So we did that. We used our favorite, the virus. It's called adenovirus. I have been working on that as a vaccine vector for a long time. Okay? That's not oncolytic, not replicating virus. Okay? So we use adenovirus to express DCT first to treat, pre-established B16 tumor for 14 days, and then we give a secondary injection VSV, our oncolytic the vaccine. So now you see that If we use add empty vector, to prime the system followed by VSV DCT, you can see that the primary response against the virus, but not transgene. However, though, if we prime the system with added DCT followed by VSV DCT, now the responses shift to anti-DCT, but not antiviral response. That response indeed translates to significantly prolonged survival and some cure, the proportion of the mice as well. So we said the oncolytic virus, the replicating virus in the tumor may also, alter the tumor environment, right, to recruit more T cells into the tumor. Indeed, we saw more antigen-specific, the T cells following VSV DCT boosting within the tumor, suggesting indeed the viral replication may create, inflammatory environment to recruit more T cells into the tumor. And also we saw T cell responses against other antigens. The antigens were not in our vaccine formulation, suggesting some antigens were spread, right, after destruction, the tumor lysis and the endogenous ABCs may pick up those tumor contents and present antigen to the immune system. So we can achieve now T cell responses not only against the vaccine antigen, but also other tumor antigens. So you may have sort of a synergy to prevent the tumor escape from just one specific attack. Now, because the time, I cannot go through the whole data, but you have to just believe me what I'm going to tell you. Byram spent 2 years trying to understand why rabidoviruses are such unique booster agent. So what he found, so we just recently submit all his story, so that we'll need a lot of hour to go through. So what he found, the key findings are here. I'm going to highlight here. So, sorry about that. So in the spleen, okay, so there's a red pulp and a white pulp. So the white pulp actually consists of follicles. Okay? So here's a diagram of the follicle surrounded by marginal zone. So within the marginal zone, there are many, macrophages and different other cells, and then blood vessels just come into the marginal zone, they open up, the blood flow will become slow in that area. So that's the normal anatomic structure to filter any blood-borne particles, right, for the system to clean up any contaminated particles in the blood. Now, the virus through IV injection get into the follicle where they infect some follicular B cells, right? Those infected follicular B cells undergo death, apoptosis. As we pointed out before, the normal cells infected by virus, they will quickly turn on their apoptotic program to kill themselves. So those, before they kill themselves, the transgenes, the antigens will get expressed in the cells. And the apoptotic B cells will, or antigens in other forms will be picked up by laboring dendritic cells, and those cells will present antigen to memory T cells, antigen specific. Because within the follicle or follicular region, there's a T cell zone. That's where naive and central memory T cells reside. So they are very, very close. The antigen carrying dendritic cells can talk to central memory T cells quickly. Now, the most important thing here is once T cells are activated to become effector cells, become killers, they will come out from the marginal zone, from the follicle to the red pulp. And once they come out, the effector cells cannot circulate back to that same region. So that's where the antigen carrying cells can keep talking to central memory cells, become a privileged area where the effector cells cannot clean them. So that's where the antigen presentation can be prolonged in that region. That's how we understand why rabidoviruses are such unique the boosters, because that's where they ended up. and how they talk to different cells in that region. So with the potency, with the understanding of the mechanisms, the next move is how we are going to translate into the clinical trial. So because I just mentioned the rabidoviruses, it's a big family, there are so many different viruses. So we wondered whether there are some other viruses in the same family actually have better capacity in both oncolysis and the immune stimulation. So David Stoyle, another collaborator who is in Ottawa, made a lot of effort to screen, the libraries to identify and to characterize and to re-engineer those rabidoviruses, and ended up with a virus called Maraba. So those are other viruses, the examples of the viruses he screened within the rabido family. Okay? So what we found actually, the Maraba virus has the best killing capacity, at least in vitro, using NCI60 cell panels. They kill more. It actually is more potent than VSV, I just mentioned. And then Stoyle went down to further manipulate the viruses by causing some mutations in the M and G two regions, and to make the viruses further attenuate to increase safety profile and still oncolytic, okay? To kill the tumor. So here's the proof, the evidence showing the increase of the safety. So the mutated virus, when we use that to infect the normal cells, right, compared to the wild type of Maraba, and there's this huge, the increase of the EC50 value, you have to, use way more the higher dose of mutated virus to kill half of the normal cells in culture. And more interestingly, actually, those mutated virus or attenuated viruses have way better capacity and to kill the tumor cells. To test that, the capacity in vivo, so we started with a xeno animal model where we using the human ovarian tumor cells and in the immune deficient the animals, where you can see the tumors can form in the peritoneal capacity. If we treat animals with the attenuated viruses, then we could see the tumor clearance quickly in a few days, and that actually can lead to, so if we lead to sort of tumor-free long-term survival. So this is a comparison between Maraba and the VSV at lower dose. So that when we use a lower dose, the VSV did not work that well, and Maraba was good. And if we increase the dose, and with VSV still, better, but did not give it the cure, and the Maraba actually gave way much more efficacy. So then we look at in immune-competent animals using syngeneic model to see whether in the contact of immune system, the viruses still work as oncolytic virus well enough. So this is the B6, again, CD26 lung, colon carcinoma, but we IV injected to foreign lung metastasis. So you can see that after the viral treatment, the tumor cells were cleared, and the animals actually survived a long time. So finally, we want to see whether Maraba is similar to VSV, can also function as oncolytic vaccine vector, can stimulate immune response in addition to its oncolytic effect. So we engineered Maraba virus to express DCT, the same antigen, so we can compare with the VSV. So you can see that we use two different, pretty aggressive, challenging models. One is lung metastasis, one is brain metastasis. And you can see that the Maraba virus as a booster can, generate prolonged survival and some cures as well. So we decide Maraba is our clinical candidate for trial. Now we need to decide on what antigen we want to use to target. So after, again, intensive discussion involved several groups, we decided on MAGE-A3. So MAGE-A3 is testis, the antigen that is only restrictedly expressed in the testis, right? But once you have a transformation, the malignancy, the MAGE-A3 will be overexpressed. So it's being found in several different types of tumors, and the MAGE-A3 is overly expressed. So we decided to target the MAGE-A3. We'll put MAGE-A3 into both adenovirus and Maraba and we produced clinical grade viruses. One is at MAC and one is in Ottawa. So now we got all clinical virus released. Then Health Canada asked us to test the toxicity in larger animals, right, before we start to do the trial. So that's what we did. We used 28 monkeys, and what we found, actually, there's almost no dose limited toxicity. We used actually 2 times 10 to the 11th so lot the PFU of the virus in monkeys. We didn't see any severe toxicity. There's some flu transit, flu-like symptoms, fever, and recovered quickly. And we did not see any viral persistence in any organs either. And in terms of immune responses, it's pretty impressive because those are the large animals, and that we actually first tested the prime boost. So we can see, all those monkeys respond to the prime boost, and the highest response could achieve the 2-digit percent of the responses we were ever seeing in large animals or human patients. And we also thought these responses could be long-lasting. In several months, we still saw the high-level response in those monkeys. So now we have sort of our phase 1 trial proved that the design was each vector alone. We have to test it first. And we decided to inject Maraba virus twice, 2 days apart, and add just once. And so now we have to test those 2 viruses separately before we can put them together as a prime boost, the vector. So now trial is already open. We have so far, I think we probably have treated more than 4 patients, and we give 1 times 10 to the 10th PFU twice into those patients. They seem to be pretty safe. We aim at sort of the prime boost testing in this summer, if we can quickly finish the virus testing in patients. So finally, we also think this approach might be broadly useful, not only for human patients, it's also for pets. And we also initiate another trial in collaboration with a few individuals at the University of Guelph to test this prime boost approach in cats with memory carcinoma. So the idea here is this is not animal models. These are patients, as we heard this morning. And if it works, it could be the pet medicine. And also we can learn a lot from this trial or similar trials in cats and dogs because they have spontaneous tumors which are very similar to those in humans by aggressiveness, by pathology. So we think, this may need to, some the real therapies for pets, and also it's a stepping stone trial to learn, everything for human trial as well. Because this is a trial. So this the trial, because the conventional therapy is the first choice, the surgery. So we cannot, ethically, we cannot bypass that. So it's different from our human trial. So we'll do the first priming vaccine before the surgery, and then we'll wait for a month after surgery and give a second Maraba, the boosting. So this trial is currently ongoing. So, oncologic viral therapy, immunotherapy, chemotherapy, radiotherapy, right? And so all targeting or small molecule targeting therapy. I personally don't believe any of those therapies alone will solve the whole problem. So the combination therapy is clearly the way for the future. So we think our oncolytic vaccine will offer a very potent and a flexible platform to combine with other immunotherapies, such as conventional vaccine therapy, the ACT, adaptive T cell therapy, and even the antibody, the H checkpoint inhibitor therapies, because all those therapies are rely on antigen-specific CD8 T cells, and the best our oncolytic vaccine can do is to expand, the CD8 T cells and recruit them into the tumor to the site to do the job, and possibly because of the inflammatory responses or inflammatory environment caused by viral replication within the tumor, which can also sustain the T cell activity to overcome the immunosuppressive the environment. So this is the group work involved a lot of investigators at the MAC and Guelph here as well, and otherwise, especially John Bell made a lot of effort to push this into phase 1 trial. Brian Lickety also, he's the leader of the CAT trial, and he made a lot of effort to do that as well. And then David Stoyle discovered the Marburg viruses, did a lot of the work, and Andrew and you have the monkey work was over there. Thanks. Take a question.
Questions? We'll try and repeat them for the live broadcast.
Right. So the question was whether we have a look at the inflammatory components, the cellular components in the tumor following this oncolytic vaccine therapy. Actually, we did. I don't have time to present the data. Interestingly, this rabidovirus infection in the tumor did not necessarily remove myeloid suppressive cells, but they convert those myeloid cells from immunosuppressive cells to actually immunostimulating and even cytolytic. The fact we have data showing that. In terms of lymphoid regulatory cells, because this virus is type of interferon sensitive, and also is type of interferon inducing. So they induce a lot of type of interferon immediately in few hours after inoculation. That causes lymphopenia. So as a consequence, a lot of cells, including regulatory T cells, were removed.
All right, thank you very much.